All right, let's continue. So, how do we regulate this contraction? Well, that regulation is going to start at the neuromuscular junction. As we mentioned in previous audios, the neuromuscular junction is where a motor neuron, the axon terminal of a singular motor neuron, comes in contact with a muscle fiber. This contact is between the axon terminal and the motor end plate on the muscle fiber. And this is where we have that exchange of the action potential. This junction is also referred to generically as a synapse. The synapse is going to be composed of a singular point between any two cells, but most specifically a nerve cell and another cell, where there is communication across a small gap. That gap itself is going to be referred to as the synaptic cleft. And the thing that is going to do the communication is neurotransmitters. Those neurotransmitters are stored in synaptic vesicles in the axon terminal of the motor neuron and are going to bind with the plasma membrane of the neuron, releasing their neurotransmitters, whatever that one may be, into this synaptic cleft, and those neurotransmitters will then diffuse across the gap to the receptors on the motor end plate. Motor end plate here because we're talking about a neuromuscular junction. In particular, because we are talking about a neuromuscular junction, the neurotransmitter in question is going to be acetylcholine. Acetylcholine is abbreviated ACH and is a very, very, very important neurotransmitter. We have a whole slew of medications that impact acetylcholine in a variety of different ways. And so ACH is an abbreviation that you really need to get comfortable with if you aren't already. So ACH is going to be released by the axon terminal of the neuron, motor neuron specifically, and it is going to float across the synaptic cleft and bind to acetylcholine receptors that are located on the motor end plate, which is located on the muscle fiber. These receptors are going to bind ACH. This is going to generate an action potential inside of the muscle fiber. Specifically, it's going to create this action potential because the binding of acetylcholine is going to open sodium channels. These sodium channels are going to open, allowing sodium to move out of the cell, into the cell. Potassium is actually going to move out. Sodium is going to move in. This is going to cause the cell membrane, muscle fiber cell membrane, to depolarize, and that's going to generate this action potential. Now, we will actually talk about in more detail depolarization, repolarization, hyperpolarization, and what all those terms mean. We'll talk about synaptic clefts and spaces and vesicles and axon terminals and all those fun things in far more detail in the nervous system. All right. So, this action potential is going to be transmitted over the entire sarcolemma surface, so over the entire muscle fiber surface, and down into every single T-tubule. This is this excitation-contraction coupling phase. So, action potential is generated because sodium floods in because of the binding of acetylcholine, which opens sodium channels. This action potential is going to go over the sarcolemma or cell plasma membrane and also travel down the T-tubules, which are stationed at the ends of every single sarcolemma. This change in the voltage is going to trigger the release of calcium from the sarcoplasmic reticulum that is located on either side of the T-tubule in the lateral sacs. This calcium, which they were storing, is going to be released into the sarcoplasm. This calcium, in turn, is going to bind to that troponin, as we saw in the last audio. The troponin is going to shift, exposing the tropomyosin. The troponin is going to cause the tropomyosin to shift, exposing the myosin binding sites, and myosin is going to bind, and we're going to get the cross-bridge cycling. This is going to cause the thin and thick filaments to move over each other, causing the muscle to shorten. Now, regulating the level of calcium inside the muscle cells is going to be up to two compounds, calcicrestrian and calmodulin. Now, acetylcholine is released into the synaptic cleft, and if it is left there, it will continuously open the sodium channels. So we need a way to break it down. There's an enzyme known as acetylcholine esterase, and this enzyme will rapidly break down acetylcholine that is located in that neuromuscular junction. When this acetylcholine is broken down, it cannot continuously bind, which means we will have a limited action potential. We will not have continuous excitation, unless there is something that interferes with that breakdown. And there are actually a variety of drugs and diseases that will interfere with the events in the neuromuscular junction. For instance, if you have an autoimmune disease known as myasthenia gravis, you will actually have a loss of function of the acetylcholine receptors. So very little acetylcholine will bind before the acetylcholine esterase destroys it all, thereby inhibiting the action potential altogether. There is also a poison arrow frog toxin known as curare, and this binds irreversibly to and blocks those receptors. When something binds irreversibly, it means it does not matter what you do. It will never, ever, ever let go. So, the only way to get rid of this irreversible binding is to replace the cell in question. Which with some cells never happens. Your nerve cells, for instance, very, very, very rarely will they be replaced in your lifetime. So, and when it binds, it blocks the acetylcholine receptors, meaning acetylcholine cannot bind, meaning the sodium channels cannot open. Which means we can't have an action potential, which means we can't have a contraction. All right. Moving into muscle metabolism. So muscle metabolism is just like all metabolism. It's all about energy availability. In any given moment, there's actually not a lot of energy available. However, energy needs to be available for cross-bridging and calcium removal. So, we're going to replenish ATP as it is used. Remember, ATP is our energy molecule in a couple of different ways. There's something known as a creatinine phosphate energy storage system. There's something known as anaerobic glycolysis and the lactate acid system, and aerobic respiration. All right, direct phosphorylation creatinine phosphate system, also known as CRP. This is going to be stored within the cell, and it allows for rapid ATP replenishment. Only a little bit of this creatinine phosphate is available at any given moment, about 10 to 30 seconds worth of energy, and then it has to be restored. This creatinine phosphate has to be replenished by the bloodstream. So this is very fast, quick. This is like a 30-second sprint. Anaerobic glycolysis, however, is the aerobic glycolysis just without the presence of oxygen. So anaerobic, remember, refers to no oxygen present. So a glucose molecule goes into glycolysis, and when it that process is done, instead of going into the citric acid cycle, it instead is going to get converted into lactic acid. This does not create additional energy, this lactic acid process. So we are limited to the amount of energy that is created by glycolysis, which is 2 ATP. This is also typically only really useful for short-term situations, think 30 seconds to a minute. Part of the reason for this is because lactic acid is a byproduct, and that lactic acid is going to build up, further inhibiting the glycolysis cycle. And then we have our aerobic system. This is our major energy producing system. It does require oxygen, though, in order for it to occur. So if you're not bringing enough oxygen to your muscles, you will not have this happen. So oxygen is actually going to get there in two different ways. It's going to come in via your red blood cells in your bloodstream, and you will have some stored in the form of a myoglobin storage depot. Now, glycolysis, it's going to use multiple different sources or substrates to create this energy. Carbohydrates, lipids, and proteins are all perfectly valid. The most efficient one, and the one this system likes to use, is glucose. Glucose creates 38 ATP and fits right into this system, easy peasy. However, you can put fatty acids in. However, in order for them to go into this cycle, other proteins or other enzymes are going to be involved, other processes, in order to break them down and convert them into a format that will go into this process, specifically into the citric acid cycle for the most part. Same thing with proteins. They need to be broken apart first and then converted into a format that can go into the aerobic respiration system, aerobic metabolism, depending on your textbook. And again, it takes longer. However, as long as we have a steady stream of oxygen, this is ideal for long-term exercise, which is why if you have an endurance athlete, you are going to see them working on improving their respiratory capabilities through long runs. They're going to build up their distance, which helps decrease muscle fatigue, but they're also at that same time increasing their lung capacity and their lungs' ability to work at that higher level for longer. So it's not just the skeletal muscles that are being worked. If you try and run, let's say 13.1 miles without doing that buildup, you're going to run out of your lungs' capabilities of bringing in oxygen relatively quickly, which means you are going to tap into your myoglobin stores relatively quickly, and usually within about, depending on your base level, 15 minutes tops. You're out. You're done. Oxygen stores are depleted. You have depleted your CRP and anaerobic respiration system, and you're going to be struggling with what little your lungs are bringing in because they are not used to it. And this particular diagram is helping to summarize these 3 steps. If we are using the anaerobic version of this, we go through glycolysis, we then convert the end product into lactic acid. This is going to create something that is referred to as an oxygen debt. Oxygen debt is the amount of oxygen that is needed to restore your muscle tissue, and for that matter, the rest of your body, to its pre-exercise state. So we have restored our ATP levels, we have restored our stored oxygen levels, we have restored our creatine phosphate levels, we have restored our glycogen, which is our stored glucose levels, and pH has returned to normal, especially in the muscle tissues. And this means that the lactic acid has been converted and or recycled back into glucose by the liver. In order for lactic acid to be converted back into glucose by the liver, it requires oxygen. Oxygen is a component of this process. This is why this does not occur during the exercise itself. So depending on how much lactic acid you have built up, this oxygen debt could go on for several days. Meaning until, if you, let's say you go for a 13.1 mile run, and then you decide to go for a run the next day, you're going to be a hurting puppy because you are starting in the negative when it comes to oxygen. There is very little chance you have gotten out of your oxygen debt from your 13.1 mile run. Which is why a lot of times they encourage rest days. Rest days give your body a chance to get back to the steady state, back to where it wants to be. All right, and then just to, no, we're going to talk about this with the next audio.